So let me start the recording. Okay, so antibiotics. Like I said, extensive lecture, fairly extensive lecture, but as always, we can only scratch the surface of such a vast topic. There's so much discovered about the antibiotics, and the antibiotics is the last class of antimicrobials that we will look at in the scope of this course. So we looked at preservatives a little bit. We looked at disinfectants, and then we're going to look at the antibiotics. So very quick overview of these of the lecture. We will talk about some general information, some synthesis aspects of about the antibiotics. We'll talk about the spectrum of activity of the antibiotics, and then we will talk about around 5, 6 different classes of antibiotics, depending on their mode of action. So different classes will have different mode of action. So we will study those. We will talk about some specific representatives, some interesting players within each group, and so on and so forth. Modes of resistance to antibiotics will be covered in the next lecture, which we will probably start in the second half, I guess, of next week's lecture. And we'll talk about how bacteria basically develops resistance against antibiotics. So a lot of things that we need to talk about. Sources of antibiotics. I guess let's very briefly talk about, before we talk about the sources, we'll talk about a little bit of history of antibiotics. Very briefly, I already mentioned this to you when we had the first couple of lectures for this course. So the antibiotics were discovered at the start of the 20th century. Penicillin was not the first antibiotic, a very common misconception that was discovered. Sulfa drugs were the first ones that were discovered and used, for the general public. They're still being employed, the sulfa drugs, the sulfa antibiotics, sulfonamides. Penicillin was the first one that was produced in large scale, properly purified, properly administered, and the one that had the most impact on the society, so to say. I told you before, it's a very, very interesting. Antibiotics were the game changer for society in a way. Back at the start of the 20th century, and obviously prior to that, people had absolutely no idea how to treat infectious diseases, especially the serious cases. They just had a bunch of folk medicines and some really dangerous chemicals that in some cases had bactericidal effect and could help with infection, but really were very dangerous for human health. So antibiotics, once people discovered it, once they realized each infectious disease is caused by some sort of a bacteria, well, some cases I guess fungi, but antibiotics specifically, we're going to be talking about the chemicals that act on bacteria. Very common misconception. Some people think that antibiotics are also good against fungi. No, antibiotics are specifically the chemicals against bacteria. The way they work, the way they kill bacteria, the way they inhibit bacteria, they rely on bacterial physiology. So when people discovered those antibiotics, the first ones, once they realized what is the principle behind the mode of activity of antibiotics, there was a huge uplift in the scientific circles and medical circles. People were super happy because back in those days, infectious diseases were #1 cause of death in the society in developing countries, and even in developed countries, and even more so in developing world. Like I told you before, you go out on a rainy day, you pick up some sore throat, you have nothing. You do have some folk medicine and just general type of medicines, but you don't have any antibiotics. And if that sore throat becomes really bad, you basically, your chances are like 50-50 whether you're going to survive or not. It's not like you go to a doctor, he prescribes you ciprofloxacin or something and you're cured within a week. No such thing. So infectious diseases were the most common cause of death. Cancer and everything that we are concerned about these days was nothing compared to infectious diseases. Not like people were not dying, but they were obviously, but because of not only because of infectious diseases, but predominantly, I would imagine, uh, because of the infectious diseases, people were just not getting to that age where cancer would be such a bad problem. Again, not to say that people were not dying from cancer. Obviously, there are unfortunately young people dying from cancer, so it can happen at any point in time. But you get the idea, right? Overall, the average life expectancy was not as high for multiple other reasons as well. Obviously, quality of life in general, and so on and so forth. So infectious diseases were the biggest problem. Once antibiotics were discovered, people were extremely happy. People thought that now we can conquer, the infectious diseases, eradicate them completely. And you probably heard this multiple times. You've read about it probably in the books. It was claimed actually in, can't remember the exact year, not that important, in late 50s, I think it was, or early 60s, WHO, one of the representatives of WHO, World Health Organization, claimed that we defeated all infectious diseases. That was a very famous claim. So he said that 60 years ago, more than 60 years ago. And as you know, it doesn't look like we defeated anything, right? Um, Not to say that we made a huge progress fighting infectious diseases. Obviously, we did, but to say that we defeated them is obviously a huge overstatement. And that has to do with resistance towards antibiotics, which we will cover next week. But the point I'm trying to bring home is infectious diseases and the agents that cause them, the bacteria, are constantly evolving. They're constantly changing, getting better at fighting back. Uh, so to claim that we defeated I don't even know if we ever will be able to defeat the infectious diseases unless we made some huge breakthroughs and some conceptually, it's possible, I guess, in the future. I mean, anything is possible nowadays with all the technology, nanomedicine and whatnot. Who knows? But the point being is bacteria is abundant. I told you before, I'm pretty sure I told you before, fun fact, our bodies are made of mostly of bacterial cells. I think I told you about it before. If you were to decompose a human body into single cells, kind of like have two piles of cells, eukaryotic cells, human cells to the left, so to say, bacterial cells to the right. If you were to do so, you would find that about 70% of all the cells, I mean, that value fluctuates. Last time when I was checking that I remember it was roughly like 65-70% of all the cells in our body are actually bacterial cells and not human cells. So if you will, we are more of a bacteria than actually people. If you were to approach a human being from like purely quantitative perspective, you would have to say that we are mostly bacteria than we are humans. That's just facts. So, not to mention that bacteria is abundant in soil, I mean, all sorts of media everywhere, water, oceans, and whatnot. So, bacteria is constantly evolving. And as last 100 years, roughly 100 years, showed, as smart as humans and with all their science, people with all their science can be, we just don't have enough resources sometimes to keep up with the evolution of bacteria. And therefore infectious diseases could be eradicated, or some of them could be eradicated for some time, but they are almost inevitably making their way back, as we are seeing these days. I think you should have probably seen it in the news. Every so often they kind of drop this kind of scary statement on you in the news, in the talk shows, that people, the scientists in this field, in the antibiotics field, in the microbiology in general, they claim that the 21st century is going to be dark ages for us with respect to infectious diseases. So they are expecting a resurrection, if you will, of a lot of infectious diseases that were seemingly eradicated in the 20th century, and we are seeing that already, like chickenpox, a lot of different diseases like that that were claimed to be eradicated in the civilized world. I mean, we're not talking about natural reservoirs and stuff, but in major countries, major cities have been eradicated, major populations have been eradicated. So now they are seemingly coming back, and people are reporting outbreaks of these diseases more and more. So that's just highlights the importance of antibiotics. If anything, in 20 for the 21st century, antibiotics are probably going to be well, that's not the right statement to make, but um, we are, I guess, in more in our need for antibiotics is much higher than it was in the past, and much higher than it ever was in the past 70, 60 years, 60 to 70 years. Once people started mass producing antibiotics, like roughly in 50s, 60s, we were fine. And then bacteria started mutating, developing resistance, and then we started losing this race against bacteria. So now in 2024, we are kind of I don't want to say we lost the race. We definitely didn't lose the race. I mean, people don't die uh, to simple infections the way they used to 100 years ago. Definitely not. But there are definitely a lot of pressing issues that we will be talking about in this lecture, and a lot of interesting aspects behind the antibiotics research and why we are in the trouble that we are. There's multiple reasons for it. It's not just because of the evolution of bacteria. All right, so this is just like a little bit of a kind of overview for the antibiotics to give you an idea how important the antibiotic. Now with all the COVID showed it, right? Even though it's a virus, obviously, but with people mass traveling back and forth across the continents. So any infection, whether it's a viral or bacterial infection, it can be transmitted between the continents, between the civilizations, if you will, right? Different corners of the world, super quickly, super quickly. All right, so as far as the origins, the sources rather of antibiotics, so where the antibiotics are coming from, there are three, I guess, major sources of antibiotics. Natural sources, which are the microorganisms, fungi and bacteria. Some bacteria-specific classes of bacteria, specific genera of bacteria are very, very good at synthesizing antibiotics, as well as some fungi. For example, penicillin was isolated from fungi, as you know. Nowadays, I would say, I'm pretty sure, not maybe 100%, but like 95%, I'm sure that yeah, pretty much 100%. I think bacteria is a much bigger source of antibiotics than fungi. So AB here doesn't necessarily mean that fungi are the biggest source of antibiotics. No, bacteria is. Bacteria is. So the natural source is microorganisms. But we also can obviously synthesize the antibiotics from scratch by using the means of chemical synthesis, right, in the lab. Surprisingly, this is not a very reliable, I guess, source of antibiotics. So make a note of that if you're taking notes. This is surprisingly actually not the biggest source of antibiotics. The synthesis, pure synthesis. Why is that the case? First of all, it's still very expensive to synthesize antibiotics from scratch. Maybe even the bigger reason, I don't know, probably even a bigger reason, we still are unable to synthesize a lot of antibiotics in its complexity, the way the nature has created them. We still rely on isolating a lot of antibiotics from microorganisms because fungi and bacteria can synthesize those complex structures much better than we can. Okay, that applies to most antibiotics, not all of them. There are some very simple antibiotics in terms of their structure, but most antibiotics actually still are, I don't know, yeah, they're still very complicated to synthesize in the lab. Because of all different chemical aspects of the molecules, we're still unable to. So synthetic antibiotics are not that predominant. They're not that common. Semi-synthesis, this might be a new word for you, even though I think I mentioned this in the earlier lecture. Semi-synthesis is the process when you take the backbone of antibiotic synthesized by the natural sources, so fungi or bacteria. So you take that backbone and then you slightly tweak it with your means of chemical synthesis. So you don't produce that antibiotic from scratch, right? When you mix things in the test tube, so to say, from scratch, but you actually take that nature already has for us, and then you slightly modify it here and there. You add a few functional groups, you remove some functional groups, and so on and so forth. Uh, that way you can make antibiotics more effective, less effective, less toxic to people, more toxic to bacteria, more water soluble, less water soluble, and so on and so forth. So you can modify them significantly. And semi-synthesis is actually extremely common source of antibiotics these days. Probably the biggest source of antibiotics, new antibiotics these days, even bigger than the natural sources. Why is that the case? A couple of reasons. Um, the first one, I guess, would be we are kind of exhausting the nature. The antibiotics coming from nature. It's not that easy to find an antibiotic in nature that would I guess follow all our criteria, right? All the easy ones we already found them, right? We already found them. We already purified them. We already been using them for decades and decades. The new ones are not that easy to find. So it's a lot easier to take an antibiotic that has been already found and then start playing with it chemically, right? A little bit, modifying it a little bit. Um, Another additional, I guess, benefit of this approach is that when you take an antibiotic that has been already identified, that has been already used, let's say penicillin or let's say vancomycin, and then you start modifying it using the means of semisynthesis. Uh, it is much easier, much cheaper, and much faster to gain the approval of the regulatory agency for that kind of antibiotic, as opposed to gaining approval for the brand new antibiotic, because the brand new antibiotic is, as the name suggests, brand new. Nobody's seen it before. Nobody used it before. People were never administered that antibiotic before. So the regulatory agency, Health Canada, FDA, whatever, they have no idea how toxic this antibiotic could be to people. So they require extensive testing. And that is obviously money, that is obviously time. If you take already antibiotic that people were using for decades, let's say vancomycin, and you start playing around a little bit chemically with its functional groups, even though you still have to go through some key safety testing, but the backbone, the working end, if you will, the main, I guess, I don't know, skeleton of the molecule has been already tested for safety before. So it's much easier to gain approval uh, for for this kind of antibiotic. Okay. That means it's easier for the labs to come up with these antibiotics, and it's easier for labs to gain approval for these antibiotics. And that is a huge factor. Everything, when we talk about the antibiotics, as I will show you later, as I will talk about it later, um, any research, antibiotics is not an exception. Uh, Money is a big topic. How much something costs, how much effort it takes, how much profit it gives back is a huge uh, point these days. Because everything now is made in vast volumes. That means lots of investment from the pharmaceutical companies, and if the profit that comes back is insufficient, pharmaceutical company will never proceed, with that kind of a drug, just because they're losing money and, they're losing money, right? They're not going to be pursuing dozens and dozens of novel antibiotics if they're losing money on every single one of them. And jumping ahead a little bit without any details, the biggest problem with lack of novel antibiotics these days is because antibiotics are not profitable enough for pharmaceutical companies to invest in them. They're one of the least out of the major classes of drugs, antibiotics are one of the least profitable drugs. Uh, we'll talk about it more later, okay? But this is just something to keep in mind. So semi-synthesis that cuts on a lot of those expenses is a very, I guess, attractive source of novel antibiotics for researchers and for the labs. And the most money that gets pumped into antibiotics research gets pumped into semi-synthesis, gets invested into semi-synthesis. Because people know this is a much faster, much easier, much better way of finding new antibiotics and eventually not just finding new antibiotics, but also successfully putting them out on the market shelves, right, on the pharmacy shelves. All right, so we talked so much. I already mentioned so much about antibiotics, but people might wonder. Well, hopefully everybody knows what they are, but some core definitions. So, what is an antibiotic? Or what is this antimicrobial that we call antibiotic? So, antibiotic substance doesn't necessarily have to be produced by a microorganism, but most of the time it is. But it inhibits or kills another microorganism. Could be inhibiting fungi, could be inhibiting bacteria. Okay. So keep that in mind. It doesn't always inhibit, it can also kill. Different people have different misconceptions. Some people tend to think that antibiotics always kill bacteria. Some people tend to think that antibiotics always inhibit the bacteria. And it can be both. It can be bactericidal antibiotic, it can be bacteriostatic. We'll talk about that later. Okay, antibiotics do not include antivirals and antifungals. Don't confuse it. A lot of misconception. Antibiotics specifically target bacteria and nothing else. I've seen it in my life. I'm sure some of you might have seen it as well, might have encountered this in your experience. I've seen people in my life that were taking antibiotics for flu, like literally thinking that it will help them defeat flu. And some bizarre cases, will remember this for the rest of my life. I knew somebody who was taking antibiotic, ampicillin specifically, for the headache. That was just hilarious, as you can imagine. But the person was taking antibiotics, ampicillin for the headache. And when I asked, why are you doing this? What are you supposed to get out of it? The person said, I don't know. My parents told me ever since I knew myself that, that ampicillin helps against headaches. So that was just, I mean, it's hilarious, but at the same time, it's pretty scary, right? Antibiotics are pretty serious drugs. You don't want to be taking them on a regular basis against headaches. Uh, I mean, they're not going to help you with that. So a lot of issues like that, and there's some more issues. A lot of as you to generalize this statement, I guess to generalize this subject, antibiotics are very common. The targets for the misuse. The people are misusing antibiotics quite often, and in a lot of countries, not in every country, but a lot of countries. I think nowadays it's like most of the major countries actually you require a subscription to get an antibiotic from the pharmacy. Back in the day, it wasn't the case. You could get some at least simpler antibiotics like ampicillin, I don't know, penicillin, erythromycin or something. You could get them without prescription. I'm sure it's still the case in some countries as well. I think some people were telling me, the students were telling me that you can still get some antibiotics, without any prescription in different countries. So, I mean, different things happen, and this actually contributes to resistance to antibiotics when people misuse them. But we'll talk about it more. But know what they are. They are specifically chemicals that are designed by nature, I guess, to kill or inhibit bacteria. And going back to microorganisms being the common source, or used to be the common source of antibiotics, you may wonder, so what do the fungi and bacteria utilize the antibiotics for when they produce them in nature? And they use them, actually, you won't believe it, but they use them for exactly the same purpose as we do. They use them to uh, inhibit other bacteria and other, well, with fungi, it's the same thing. They use it to inhibit other bacteria that they are competing with. So in the nature, fungi and bacteria producing antibiotics to compete, to eliminate the competition from other bacteria. It's just that the antibiotics are being made in nature at lower concentrations, obviously, but it still works. It inhibits bacteria that lives next to these antibiotic producing bacteria. So they serve the same role. There's other roles for the antibiotics in nature, but we'll just concentrate. Some people claim, I mean, it is not just a claim, it is also supported by facts. Antibiotics are also being utilized by bacteria as so-called carbon sinks. They just dump extra carbon from their biochemistry into antibiotics because the antibiotics have pretty complicated structure, complex structure. They have a lot of carbon atoms usually, and they use them as carbon sinks to get rid of extra carbon. So that's the common, also evolutionary, I guess, purpose of antibiotics. All right, so what are some of the properties of antibiotics, right? When we talk about antibiotics, what exactly do we imply? Antibiotics, obviously, are the chemicals or the drugs that are being administered to people, and people take them. They take them inside of their bodies, so to say, right? They swallow them, or there's a lot of antibiotics can be also administered IV or intramuscular injections can be done. So you obviously have to make sure that the antibiotics have a so-called property that is a property that is called selective toxicity. Selective toxicity. Pretty straightforward concept. I'm sure everybody knows what it is in the first place. So selective toxicity means that the antibiotics are selectively toxic. That means they're toxic only to bacteria and they're not toxic to us. Okay, for obvious reasons, because if antibiotics were toxic to people, then you would be killing people. So that means something to keep in mind. The selective toxicity has its roots in molecular biology. So there's something about the mechanism of action of antibiotics that makes them target bacteria and not target humans, make them target bacterial cells and not target human cells. So there's something particular about the way they act. And we will talk about those details, about those mechanisms. So all antibiotics that we're using are targeting specifically bacterial structures or some unique aspects of bacterial biochemistry, the ones that we don't have. So that is the molecular foundation, I guess, behind the selective toxicity. Some may say, okay, well, it is known that antibiotics can be also toxic to people. People can even die from antibiotics if misused, if abused, and so on and so forth. So that is obviously the case, as is the case with any drug, with any active medicinal compound. Um, some antibiotics can be very toxic to our kidneys and to our liver, specifically these two organs. Not only these two, we will talk about some other ones as well, but generally speaking, liver and kidneys are usually the organs that are taking the most damage from any drugs, not just antibiotics, but antibiotics as well. And there's an obvious explanation to it. Liver is the organ where that metabolizes antibiotics and other drugs. Kidneys is the organ that is responsible for excretion of drugs. So if somebody is abusing any antibiotics or the dosage is off, those two organs, for those reasons, will take the first hit, will always take the first hit because of the concentration and because of the functionality of these organs. So usually kidneys and liver, but also other organs can be affected. Depends on concentration, depends on also the health state, I guess, of individual. But fundamental property of all the antibiotics is their selective toxicity. They always target They always target bacterial structures, and they don't target human structures within the cells. Okay. It's not just the bacterial cell wall, as it says here. It's just one of the examples. There's other antibiotics that target other organelles. Um, Toxicity to host, that means to us, to people, like I already mentioned, can also happen, but these are side effects, and these are not because of antibiotics specifically acting on our organelles. It's because of the accumulation effect, because of liver or kidneys unable to deal with excessive antibiotic, for example, in your bloodstream or somebody's bloodstream. So that can also happen. We talked about the penicillins. I don't want to, I mean, the semisynthesis. I don't want to talk about it too again. You know what it is. No, make sure you know what it is for the test. Many antibiotics these days, not just these three classes. Uh, I don't know. There are probably some exceptions, but it wouldn't be a mistake to say that every single class of antibiotics these days has semi-synthetic antibiotics as well. Like I said, it's the most invested in um, field of research in antibiotics because it's for all those reasons we discussed earlier. Um, so the benefits, I also told you what are the benefits of the semisynthesis. All sorts of benefits, right? You can make the molecule less toxic to people. You can have fewer side effects than the original molecule. You can make the molecule, the antibiotic, to have a broader spectrum. We will talk about this in a second than the original molecule. You can make the molecule be able to withstand stomach acid, so low pH environment, and which will allow the doctors to administer this antibiotic orally, right? Which otherwise would probably have the molecule decomposed. Antibiotic would get decomposed if it is stable to stomach acid. If you modified it chemically in such a way that it is stable to stomach acid, it will be stable in the stomach, and therefore you will be able to administer the antibiotic orally. It's always easier to administer things orally. Um, we'll talk about the resistance. I mean, some of these modifications that are introduced by semisynthesis make the antibiotics more resistant to bacterial enzymes. Okay, that has to do with the whole topic of antimicrobial resistance, antibiotic rather resistance in bacterial species, but making the drug less susceptible to bacterial enzymes help us overcome these resistance issues. So bacteria tries to survive by mutating, and we modify the antibiotics in such a way that they are keeping their cidal or bacteriostatic potential against bacteria. And lastly, obviously, last not least, but you can modify the drug to be more efficient against specific bacteria. Vancomycin is a very good example here. Very common drug, very effective drug, and it's being constantly subject to semisynthetic modifications. Since it's a very important antibiotic, as we will talk in a few minutes. People invest a lot of money into semisynthesis of vancomycin to make sure the drug becomes relevant and stays relevant for us, because bacteria mutates, becomes resistant to it, and so forth. So all sorts of beneficial modifications to the drug that makes it relevant, I guess. Speaking of the spectrum of activity, something I mentioned in the previous slide. So what is a spectrum of activity? Pretty straightforward term. A lot of you probably know what it is. Basically refers to the fact how many different genera of bacteria, how many different species of bacteria this antibiotic can target, can kill, or can inhibit. So narrow spectrum means this particular drug has a very narrow range of microorganisms, so a very narrow range of bacteria that this antibiotic can inhibit or kill, right? Only a couple of species or maybe a couple of genera, and that's it. Very narrow spectrum. Wide spectrum is, as the name suggests, the drug that is effective. Again, whether it's either cidal or static, bactericidal or bacteriostatic against multiple, numerous species and numerous genera of bacteria. Okay. Um, so as it says here, broad spectrum inhibits many kinds of pathogens. Okay. These examples here for the narrow spectrum and the broad spectrum, you don't need to memorize them. I'm not going to be asking you, is bacitracin narrow spectrum or wide spectrum? Is nystatin narrow spectrum? So you don't have to memorize this. What you need to memorize is something that is not on the slides, and I will add it now. So I mean, the question could be, what's better, narrow spectrum or broad spectrum antibiotic? Um, why do we, if one of them is better than the other, why do we still have the other kind? So here it's not a very simple question to answer, or rather, it's not that complicated question to answer. It's just there are different perspectives on it. So uh, there is a perspective of the industry, and there's a perspective of, I guess, science and medicine to this question, on this question. So from the standpoint of industry, pharmaceutical companies, they always try to go for broad spectrum drugs. Why is that the case? Because for them, it's a broad spectrum drug is a better profit. It's the drug that you design once, you create once, you produce it, right? You approve once, and then that drug is going to be target so many different diseases, or so many different pathogens, so many different microorganisms in this case. So that is amazing, right? So they understand that it will be sold a lot better than a narrow spectrum drug, and the audience, right? The number of people that will be purchasing the broad spectrum drug is going to be bigger for obvious reasons because it targets so many different conditions, so many different diseases caused by different pathogens. So, with some exceptions, I mean, there's always exceptions, but generally, pharmaceutical companies are interested in broad spectrum antibiotics. If you were to be in antibiotics research and you came up with a potential new antibiotic and you try to like sell this patent or sell this idea to a pharmaceutical company, they would be more interested if you had a broad spectrum antibiotic in mind. Okay, so that's their stance from the scientific slash medicinal medical standpoint of view. You want to be using narrow spectrum drugs more. Why is that the case? Because of the resistance. The simple, fast answer, because of the resistance. See, our broad spectrum antibiotics are good in terms of killing many different pathogens, but there's obviously a flip side to it. Every time you kill a pathogen, or every time you kill a bacteria, you promote resistance from those species. So the more species are targeted by your drug, the higher obviously is going to be the resistance profile. The more spread will be the resistance profile. Different species will be mutating in response to your drug, and therefore the chances of generating uh, antibiotic resistant mutant will be higher. Okay, not to mention that broad spectrum antibiotics, when you administer them to people, these antibiotics, since they don't differentiate much between different genera of bacteria, they can severely damage your gut microflora. As you know, it's a very important part of your immune system, gut microflora, the bacteria that lives in your gut. Broad spectrum antibiotics are obviously much, much better at wiping out, or in general, yeah, well, like not necessarily always wiping out, but in general, affecting, killing beneficial bacteria in your gut. And losing that bacteria can be a very dangerous case. A complete wipeout or significant wiping out of gut microflora can lead even to death. People suffer severe consequences. So broad spectrum antibiotics, especially when people are taking them for a long time, say uh, it was hard for the doctors to eradicate the infection, and the individual was on extended period of time, and he or she was on the broad spectrum antibiotics. Those people can have very, very severe consequences. up to, like I said, can be lethal. It's not easily treated. There are some ideas behind how to treat the loss of gut microflora, but it's a very serious condition. So like I said, from the medical, from the scientific standpoint of view, narrow spectrum antibiotics are much better because they target only specific range of microbes, so microorganisms, so bacteria, and they obviously are much less effective at promoting resistance. And they're much less effective at wiping out beneficial bacterial strains in your body, in your gut. Okay. But again, the problem is this is just one of the illustrations, which will be quite a few throughout these two lectures. One of the illustrations of how scientific stance to the problem is not necessarily like purely scientific and as well as medical is clashing with industrial pharmaceutical companies' perspective. It's not the only one. We'll be talking a lot about these. The other one I told you earlier was about when we talked about, if you remember, the antibacterial soap, right? That the triclosan that they add to the soap is there's nothing good about it. Uh, you should remember that. But nevertheless, industry finds it's very profitable, so we still have it on the market, even though scientists all over the world have been claiming it's not a good thing to do at all. So this is just another example, kind of giving you those. So the broad spectrum can promote resistance. Like I said, they can cause so-called superinfections. Make sure you know what it means. Superinfections is when you have a patient with a specific infection, and on top of that infection, he or she develops a secondary infection, so a superinfection. Quite often, this happens with Clostridium difficile, and this is because it is a very common pathogen in hospital settings. So when the patient is in the hospital for too long with some severe infection, quite often the patient obviously is exposed to a lot of antibiotics, and because Clostridium difficile is so abundant in hospitals, it's like number one bug, so to say, in hospitals. If you, God forbid, contract a Clostridium difficile infection, if you go to the doctor, most likely the first question the doctor will ask you is whether you have been admitted to hospital recently or not. So Clostridium difficile can grow on top, so to say, can develop infection on top of already existing one. So broad spectrum infections, broad spectrum antibiotics are good, very good at promoting that. And one of the reasons they do that is because they can wipe out your beneficial flora, and beneficial flora is responsible for keeping Clostridium difficile as well as other bacteria, pathogenic, opportunistic pathogens, is responsible for keeping them in check. So if you wipe out that flora, any sort of opportunistic pathogens can grow on top, so to say. They can develop the superinfections. So keep that in mind. I will definitely have a question or two about the narrow spectrum, broad spectrum, medical / scientific versus industrial approach to the issue. As far as specific classes or specific representatives, don't have to memorize. There's so many antibiotics that don't need to memorize. A little bit about natural sources of antibiotics. I told you that this used to be the biggest source of antibiotics in the early days of antibiotics research in the previous century, obviously. But eventually we exhausted all of these natural sources, and now it's extremely hard. It's still possible, but it's very tough to find a new antibiotic that would not only be just a new antibiotic, but also the antibiotic that um, fits our criteria, right? Because criteria is a lot more stringent these days than it used to be. So which microorganisms are um, known for, notorious for production of antibiotics? Different types of genera, fungi, as well as Penicillium, Cremonium, as well as bacteria. Nowadays, like I said, bacteria is a lot more common. source of antibiotics. So remember that. And out of all these genera that you see here, I'm not asking you to memorize them except for one. It's the Streptomyces species. These guys here, this genus of bacteria. So Streptomyces uh, are responsible for like 80% of all the antibiotics that we have on the market, or the backbones of is responsible for approximately 80% of the backbones of all antibiotics that we have on the market. Quite a few, as you can tell. They're just so good at it, right? And the Streptomyces species, couple of fun facts. Um, they are bacteria. This is bacteria, but for the longest time, until like late 70s of the 20th century, people thought that they were fungi because they had a very similar structure to their cells as fungi would have. And under the microscope, they looked like fungi, but eventually they realized they were just a very weird bacteria. So they are bacteria. The reason they're so good at producing antibiotics is because they are the most common. Well, I guess it's not the reason. Well, I guess you can think of it as a reason. So the thing is, they live in soil. They inhabit the soil, and in soil, you have so many different bacteria living, obviously, and fungi that it's inevitable that Streptomyces, as well as some other ones, Streptomyces is just the champions in this. They, over time, over evolution, they needed to develop some sort of a way to, like I told you earlier, to fight off all this competition in the soil, considering how heavily the soil is contaminated with all sorts of bacteria. So they eventually started making a lot of these different antibiotics. As you can see, this is the not even obviously a complete list. This is just some of the representatives. These are different species of Streptomyces, but all of them are Streptomyces genus. Again, don't have to memorize, but do remember that Streptomyces is a single genus of bacteria that is responsible for, like I said, about 75–80% of all the antibiotics that we know that are out there on the market, modified, unmodified, whatever, but it's all coming from Streptomyces, most of it. Okay, they're very slow growers. If you were to grow this on the plate, Streptomyces take like 3–4 days, 5 days to grow on the plate. Um, they like room temperature as well, so they kind of like fungi, but they are actually bacteria. So that's that. We're not going to talk too much about bacteria. I mean, about fungi and all these like different representatives. Just that general information that I gave you should be sufficient. Now we're going to switch on to classes of antibiotics, and that's where we are going to talk about 6–7 different classes, about the mode of action as well. So before we do that, let's make a quick break. It's been an hour, even more, considering I started at 6 with recombinant DNA. So let's make a 7 minutes break and come back to the lecture at 7:37. Okay. All right, we're back for another hour. So the classes of antibiotics, I mean, there are different ways of classifying the antibiotics, but here we will look at classification based on the mode of action, specifically mode of action, molecular mode of action, how the antibiotic kills bacteria, or how does it inhibit it. So that's what I refer to as classes. Some antibiotics, this is just general statements, may have overlapping spectrum of activity. So that means some antibiotics can be killing the same bacteria, obviously, and that's because the mode of action of each of these antibiotics is equally effective, right, against that bacteria. In some cases, certain antibiotics, because of their mode of action, will be unable to kill or inhibit specific bacteria, or they could be very like barely efficient against certain bacteria because that bacteria would be lacking certain structures, certain I guess, biochemical pathways in some cases that specific antibiotics are targeting. Gram negatives and gram positives, for example, um, there are antibiotics that kill both gram negatives and gram positives, and there are antibiotics that only target one, either gram negatives or gram positives. Different classes of antibiotics have different efficacy, which is kind of follow up of what I just said. They can have a different efficacy against different organisms. And again, that depends on what kind of antibiotic we're looking at. How does it exert its effect? What kind of cell component does that antibiotic target? And whether that cell component is present at all in another bacteria. We'll look at actually one of the first classes that we will look at. We'll illustrate that. Um, Different classes of antibiotics, we will discuss this more, but just a heads up, different classes of antibiotics usually are subject to different modes of resistance from the pathogens, which again, don't just blindly memorize it, make sure you understand where this is coming from. If different classes of antibiotics have different mode of action, that means they act on different organelles, they act on different biochemical, maybe pathways, then obviously, in order for the bacteria to get resistant to those different modes of actions of antibiotics, they have to develop completely different mechanisms of resistance. Yeah. Otherwise, you know, it's not going to work. In some cases, however, like the word says here, usually, in some cases, we will talk about this more next week, one mode of resistance can actually protect against a lot of different classes, if sometimes even all of the classes of antibiotics, maybe with a little bit of different efficacy, but still. But those are rare cases. We will look at them next week. So as it says here, some modes of resistance can have an overlapping profile, meaning one mode of resistance can protect against multiple different antibiotics. Okay, and multiple different, not just multiple different antibiotics, but multiple classes of antibiotics. Multiple antibiotics may mean several antibiotics within the same class. All of them could be, for example, penicillins. So that's still the same class. I'm talking about different classes, the ones that have different mode of action. All right, but we'll talk about this more as far as the resistance is concerned when we talk about the resistance next week. As far as the modes of action, the classification of antibiotics based on modes of action. So the ones that we will look at in this lecture anyway, it's not the it's not a comprehensive list, but these are some of the major antibiotics targets that antibiotics are like impacting, right? Cell wall is one of them. Protein synthesis, so the process of translation, nucleic acid synthesis, transcription, and general metabolic pathways, the biochemical pathways that lead to the production of some sort of important biomolecules. And in this case, for the general metabolic pathways, we're obviously will be talking about the pathways that are utilized by bacteria and not utilized by people. Because if people were utilizing these pathways, then antibiotics would be also interfering with these pathways in people as well, which is against the whole concept of selective toxicity. Okay. There are some also unique components of bacterial cytoplasmic membranes, specific porins, specific channels. Porins, if you don't know what it is, it's the protein through which water is going into the cell. Small molecules in water are coming into the cell. So there are some unique components that can be also targeted by the antibiotics, and those are usually narrow spectrum antibiotics. Okay, so in some cases, pathogens recognition and attachment to the host is being targeted by antibiotics. So if that is targeted, then the pathogen cannot attach to the host cell anymore, and therefore cannot exert its effect. So, but those are more exclusive, more like unique um, types of antibiotics. So the very first one, the very first class we will look at will be the antibiotics that are inhibiting the cell wall synthesis. Okay, these antibiotics are usually called beta lactams, so this should be a Greek beta lactams, and also they are quite commonly referred to as um, penicillins, just general penicillins. Why? Because the first discovered representative of this class, as you guessed, was penicillin. Penicillin, natural penicillin, discovered in 30s, was a beta lactam. So one thing here is, like I said, this B should be Greek beta. It just, I think, auto-corrected it. So these are the ones that target the cell wall synthesis. And as soon as you hear the cell wall synthesis, you must remember what it is. This will tell you a lot about these antibiotics. So cell wall is the peptidoglycan layer that is present in large quantity, right, in gram positives only, right? So the predominant targets of the beta lactams would be the gram positives. Okay, gram negatives also have a very thin peptidoglycan layer, but it's not nearly as important as it is for the gram positives. So beta lactams are mostly efficient against gram positives, especially um, the first representatives of beta lactams that were found, like natural penicillins, some of the semi-synthetic penicillins, were exclusively active against gram positives. Eventually, these guys here, these classes here, cephalosporins and carbapenems, they have been gaining. So these are, make a note of these, like highlight them or something. These two classes of antibiotics, cephalosporins and carbapenems, are one of the most important antibiotics that we have right now at our um, disposal as a medical community. So these are some of the most important ones. Um, they're one of the most efficient ones. If we lose these antibiotics to resistance, it's going to be a big we're going to have a lot of trouble. So these antibiotics, they have undergone, these two classes of antibiotics, cephalosporins and carbapenems, they have undergone multiple cycles of improvement over decades. So it's been generated, there's been generated multiple generations of these drugs, so these antibiotics. And as I will show you later, these drugs were made progressively more and more effective, not only against gram positives, but also against gram negatives. But going back to, I guess, the mechanism of action of uh, beta lactams, right, of the penicillins. The better word is beta lactam because penicillins specifically refers to a particular representative that is by now is the least utilized antibiotic out there. Nobody uses penicillin in clinics anymore. Well, maybe some exceptional cases, but penicillin, natural penicillin, is not very effective anymore because most bacteria is resistant to it. So what is the mode of action of this class of antibiotics? In order to understand that mode of action, you need to remember the structure of the peptidoglycan layer. So the peptidoglycan layer is made of these repetitive chains here on the figure in the right bottom right corner. You have these chains of sugars that are made of NAG and NAM subunits. So these kind of beads are the subunits NAG and NAM subunits. They are making up these chains of sugars. And these chains of sugars are, as you can see, cross-linked with these small stretches. And these small stretches are actually oligopeptides. They are short peptides, somewhere between 5 and 7, if I remember correctly, the length of these peptides. So they serve as literally cross-links for a very uh, uh, simple need. They are reinforcing the cell, the physical strength of the cell wall by linking these chains of NAG and NAM subunits together, as you can see, every so many residues. They are making the whole structure a lot more physically tough, which obviously results in a lot more, um, a lot better, I guess, stability against osmotic pressure for the bacterial cell. So the cell wall protects the cell, protects the gram-positive cell from any spikes of osmotic pressure inside of the cell or outside of the cell. So that's a very important structural role of the peptidoglycan layer. And again, don't forget, this is predominantly found in gram-positives. They have a very, very thick layer of peptidoglycan, um, in addition to just a single layer of cell membrane. Now, the mode of action of antibiotics is closely linked to these bridges, if you will, crosslinks that are established by the oligopeptides. And in order to see how it works, how these antibiotics work, let's you can click on this hyperlink here. Hopefully it works. Sometimes they randomly remove stuff, but I think it should be fine. I'll share the screen once I open the. Oh, that's what I was talking about. I don't think it's actually available. Okay, I'll find some better links. These links were available until like all this time. One of them, I think, for the later slide was lost eventually, but all the rest of them were working. I think they closed access to all. All right, I'll just explain in words, and then I'll link separately on eCentennial or something. The video of the mechanism, how it works, so it's a lot more visual. So it's not that complicated to understand. So what the enzymes do, what the antibiotics do, the beta lactams, what they do, they block the action of the enzymes that are responsible for establishing these crosslinks. So the enzyme that establishes the crosslink ensures that the crosslink is in place and therefore ensures the structural integrity of the cell wall. So the antibiotics, the beta lactams, they bind to those enzymes. Those enzymes are called transpeptidases. And by binding to those transpeptidases, they prevent the transpeptidases from formation of these peptide bonds between the short stretches of peptides, these oligopeptides, between the chains of NAG and NAM subunits. And obviously, if this cross-link is not established, the whole cell wall, the cell wall loses the structural integrity. It doesn't lose the structural integrity right away. It loses the structural toughness, I guess, of the peptidoglycan layer. It weakens the cell wall. And as it weakens the cell wall, any jump, any spike in osmotic pressure inside of the cell, inside of the bacterial cell, or outside of bacterial cell, will cause the cell wall to rupture. And as the cell wall ruptures, so will the cell membrane, and the whole cell will be basically leaking right into the contents of the bacterial cell will be leaking into the environment. The cell will die, and all of that will happen of the um, because of the enzymes, the transpeptidases that are responsible for establishing these crosslinks, because of those enzymes being blocked by beta lactams. That's how they act. Okay, so make sure you know the mechanism. Obviously, there will be questions on. So the cell lysis as the consequence of the weakened cell wall, there will be a cell lysis happening. Okay. One more thing, very important. I don't think it's actually a part of the slide. No, it's not. So how would the cell die? So, or let's put it this way. If I were to ask you to predict if beta lactams are um, bacteriostatic or bactericidal antibiotics, if they kill bacteria or they actually uh, um, inhibit, right, the proliferation of bacteria, what would be your answer? Uh, so most likely, majority of people will right away say, well, they obviously are bactericidal. They kill bacteria. Um, but it's actually not the case. Beta lactams are bacteriostatic, and there's a little bit of a catch here. The fact that the cell dies doesn't necessarily always mean that the antibiotic is bactericidal. I'll explain what I mean here. So think of it in this terms. Um, when will the cell die? In which cases will the cell die um, as a result of beta lactams? Which events will lead to death of the cell due to the action of beta lactams? So I already gave you one event. It's when the osmotic pressure jumps, either outside or inside of the cell. But it's only one event, fairly rare compared to the other event in bacterial life cycle. So there is something else in bacterial life cycle that happens a lot more common, which will be even more important, I guess, for from the beta lactam standpoint of view, from the mechanism of action of beta lactams. So that other event that I'm referring to is the cell replication. What does the cell replication have to do with? Because during the cell wall, once the cell has been made, once the cell has been matured, the replication of the cell wall doesn't happen. But when the cell is replicating, that's when the cell wall also replicates. And when the cell wall replicates is when these crosslinks are being established. And that means at the time of cell replication, at the time of cell wall formation, at the time of crosslinks formation, is when the cell most vulnerable to beta lactams. So it's most vulnerable to beta lactams in two cases: when the osmotic pressure jumps and when the cell replicates. And like I said, amongst these two, generally on average, usually, cell replication happens more commonly. So that means the bacterial cell, will die to beta lactams only or predominantly when it replicates, when the cell wall is being made, and so on and so forth. So, but if the cell dies to beta lactam or any other antibiotic, in this case beta lactam, only when it replicates, what kind of effect does that antibiotic have? It actually has a bacteriostatic effect, right? Because if the cell is mature and is not replicating, these antibiotics are not very effective. They will only be effective if osmotic pressure will be jumping outside or inside, but that doesn't happen much usually. I mean, it does happen, but it's not as common. Most of the time, the cell will be dying to beta lactams when it's replicating. And therefore, if replication of bacteria leads to death, that means bacteria will remain at the same concentration when exposed to beta lactam. Every time it tries to replicate, the new cells will die. So that means the antibiotic is actually bacteriostatic. It doesn't really change the concentration of existing cells in the media. It only prevents the new cells from being formed. And therefore, it prevents the numbers from increasing, and therefore it has a bacteriostatic effect. So that's a little bit of a tricky, I guess, detail that you must understand as well. So beta-lactams are actually considered to be bacteriostatic antibiotics, even though, like I said, they can also kill the cell when the osmotic pressure is spiking, both inside or outside, either or. Because once the osmotic pressure jumps, the cell will have to rely on its integrity, will have to rely in its integrity on the strength of the cell wall. But if the cell wall is compromised, but then again, this also is only going to be happening when the cell wall is getting replicated. So beta-lactams are mostly, well, not mostly, they are considered to be bacteriostatic. They are preventing from bacteria, they are preventing bacteria from increasing in numbers. So they're bacteriostatic. If the cell is not replicating, beta-lactam is unlikely to have much of an impact. Therefore, it's not really killing anything. So hopefully that's you got that. All right, so that's the mode of action pretty much. As far as specific representatives, this ring here that I'm have my cursor over, this is what is known as a beta-lactam ring, and hence the name of the antibiotics. You don't have to memorize the structure. I'm not going to be showing you the structure for you to recognize on the test, but probably just not a bad idea to generally remember what the structure looks like. We're not going to have it for every single antibiotic, just for a few. But as far as the test, you don't need to know it. What are the representatives? Some of the first ones, like I said, beta-lactams, penicillins G and V, were one of the first antibiotics employed. So the penicillins, they have barely any use these days. I mean, they do have some, I guess. Depends on the country too. Depends on financial resources and whatnot. But penicillins are not commonly employed these days because they've been used so much over the past century that most bacteria are actually having some sort of genetic element, some sort of genes that make them resistant to these antibiotics. So they have narrow spectrum of activity. I told you before that early days penicillins have a very narrow spectrum of activity, mostly against gram-positives. And they are extremely susceptible to beta-lactamases. So this is the resistance I was talking about. The beta-lactamase, we will be talking about this a lot more next week, but it doesn't hurt to get introduced to it today. Just very briefly, beta-lactamase is the enzyme. As soon as you see A's at the end, that means it's an enzyme that is cleaving the beta-lactam antibiotics. So the drug, or not the drug, the enzyme that destroys the beta-lactams. And it destroys the beta-lactams by cleaving, chemically cleaving the chemical bonds of the beta-lactam ring. So the ring becomes open and the antibiotic loses its structure, and therefore, unfortunately for us, fortunately for bacteria, it loses its efficiency. Okay, so these enzymes are called beta-lactamase is a more general name, I guess, and the penicillinase is more of a historical name because, again, same idea. Ase at the end means it's an enzyme, and the enzyme that cleaves penicillins. And the reason they were called that historically is because historically penicillins were the first beta-lactams known. So penicillins aside, like I told you, not the most efficient antibiotics these days. There is a whole bunch. That doesn't mean that the beta-lactams are not used anymore. They're actually super important antibiotics still. These are some of the representatives. And for these five, actually for these four, ampicillin, amoxicillin, imipenem, and methicillin, know them. Know that they belong to beta-lactams. I might have a question on that on the test. These are semi-synthetic beta-lactams. Also, in addition to, you know what? Let's do this. For the beta-lactams, know the imipenem, methicillin, and going back, these two classes, cephalosporins and carbapenems. Just these four. Cephalosporins, carbapenems, imipenem, and methicillin. From ampicillin, most people probably know it, but I'm not going to be asking anything about it. All you need to know for these four that I mentioned is that they belong to beta-lactams. So these are semisynthetic beta-lactams. Semisynthetic means they have broader spectrum of activity. They are more resistant to the enzymes, better absorbed by the gut. They're more stable in the acidic environment in our stomach, so they can be administered orally. And a couple more things. I told you the carbapenems and cephalosporins are later generations of these drugs, are very effective against gram negatives as well. Yeah, it was actually, let's talk about this a little bit more, and then we'll come back to the other two. So the cephalosporins act essentially the same way as penicillins, but the major benefit of them is the fact that they are a lot more resistant to penicillinases or the beta-lactamases. So these enzymes are having a lot more trouble cleaving this beta-lactam ring. It's still there, as you can see, but the enzymes have a lot tougher time breaking that beta-lactam ring than the beta-lactam ring of penicillins. And as I told you, there's been multiple generations. It's not four. By now, it is like, I can't remember now, like 7, 8 generations of them. So with each generation, as you can see here in the table, they were getting much higher spectrum of activity. And some of the later ones are even effective against gram negatives, okay, including some Pseudomonas species. So that is generally the trend for all the promising drugs, for all the promising antibiotics. They keep them on the market. They keep researching different chemical aspects of these antibiotics, and they modify them to keep them relevant for the clinical for the clinics. Okay. So the same applies to carbapenems. We're not going to talk separately about the carbapenems, but the same idea. For this table, all you have to remember is that as you go from first generation to last, you are getting drugs that are more resistant to the action of enzymes, to the action of bacterial enzymes that cleave these antibiotics. And the fact that they are becoming more and more broad spectrum, they're becoming more effective against gram negatives. So as it says, with each generation, more activity against gram negative, and I guess less activity against gram positives, but the good cephalosporins and carbapenems, they're also effective against gram positives as well. Okay. Two more drugs that I told you need to remember, imipenem and methicillin. Let's start with methicillin. Methicillin was, some of you might have heard about it, or you might have heard about it, but you never paid attention to it actually. A lot of you probably know a so-called MRSA strain. MRSA was, some 20 years ago, still relevant, I guess, but it used to be a very resistant bacteria that was given a lot of trouble in hospitals. So MRSA stands for methicillin-resistant Staph aureus. So it was this particular strain of Staph aureus that was named based on its resistance to methicillin, right? Methicillin-resistant Staph aureus. So what does that tell you? That tells you how important this antibiotic is if people are naming the whole bacteria based on its resistance to this drug. So methicillin was exceptionally effective against Staph aureus. Staph aureus is a very common gram-positive pathogen. So methicillin was very good at treating it, especially some hospital-acquired Staph aureus infections. But the MRSA, once it emerged, it was resistant to methicillin. And that gave a lot of trouble to hospitals. People were dying, and it's still like out there. It's just there are other antibiotics that we use to treat MRSA. We'll talk about them in just a second. Imipenem, the last beta-lactam that you need to remember, the fact that it belongs to beta-lactams, but also know one more property about the imipenem. It is one of the many so-called drugs of last resort. So what is the drug of last resort? As the name suggests, probably something very um, effective, but at the same time probably dangerous, and that's exactly what it is. So drugs of last resort, antibiotics of drugs last resort, are usually very effective antibiotics, but they are also very toxic to people. And the reason they're called last resort is because they are given to people only when nothing else is helping a patient. When the patient has been through uh, various antibiotics, has been on different antibiotic therapy, and nothing is helping. The infections keeps spreading, keeps proliferating. So in this case, um, when it's the question of life and death, they administer those so-called drugs of last resort. So it's very effective. Doesn't always help, but effective, but very toxic to people, to us as well. So, but there's a couple of them that we will look at. Imipenem is one of them, so make sure you remember it. Remember that it's a beta-lactam, and remember that it's a drug of last resort. Okay, the next class of, and let me try the mode of action again. Yeah, I think they closed down that website completely because this one was available as well. Okay, so I'll find you better. I'll find the better um, or alternative, I guess. Um, links to the videos so you can see how it actually looks. The animations, I guess, uh, for all these antibiotics. It's actually surprisingly tough to find a really good one. I remember when they closed the link to one of these antibiotics, aminoglycosides, I think it was, I couldn't find a good one, a short good one for the lecture purposes on the internet. But anyway, the next class we will look at are the glycopeptides. Glycopeptides. So an easy, well, I guess an easy way or one of the ways to memorize what the glycopeptides are. Glycopeptides are, in a way, similar to beta-lactams. In a way, they're not the same. It's a different class, but they also inhibit the growth of bacteria, proliferation of bacteria, by interfering, as it says here, by inhibiting the cell wall synthesis. So that the same peptidoglycan assembly is being inhibited by the glycopeptides, just like beta-lactams did. And they're just as beta-lactams, they're mostly effective against gram-positives. But glycopeptides inhibit the cell wall synthesis in a different mechanism, using a different mechanism than beta-lactams. The beta-lactams were binding to the enzymes, right? The ones that we just looked at. They were binding to the enzymes, the transpeptidases, that were cross-linking the cell wall. Glycopeptides, in their turn, they bind to the peptidoglycan precursors themselves. They bind to the NAG and NAM subunits. The peptides that are being used for the cross-linking purposes. They bind to these and they prevent the formation of the cell wall that way. So same organelle is affected, but using a different mechanism. So as you can imagine, once we discuss antibiotic resistance next week, you are fully to expect a completely different mode of resistance against glycopeptides, right, compared to the beta-lactams, and that will be the case. Different mechanism of action, different mechanism of resistance, and the other way around kind of thing. What works, the enzymes that work against beta-lactams will not work against glycopeptides. So glycopeptides are resistant to beta-lactamases because glycopeptides work differently than beta-lactams. They have a different structure. They do not have a beta-lactam ring, and therefore beta-lactamases, these enzymes, there's nothing for them to cleave. These enzymes specifically cleave the chemical bonds of the beta-lactam ring. If the ring is absent, beta-lactams are useless. So beta-lactamases are not active on glycopeptides, and therefore glycopeptides are resistant to those. The only representative of glycopeptides that you will have to memorize for the test will be the vancomycin. I already mentioned this drug before. Fairly narrow spectrum drug, also considered to be drug of last resort, and a little bit of a connection to what we already discussed. Vancomycin was that drug that helped people fight off the MRSA strains, methicillin-resistant Staph aureus, as well as enterococci. So once these strains emerged and methicillin became somewhat useless, vancomycin saved the day, so to say. Okay, and it is fairly toxic to people, so it's considered to be a drug of last resort. But I also mentioned earlier, nowadays vancomycin gains a lot of research attention. People are trying to modify it in different ways to keep it relevant for clinical applications, for hospital applications. So vancomycin, there's a lot of work done on this drug. It's a um, very common drug, I guess, very common, especially for research applications. Okay, so that's as much as you need to remember about the glycopeptides. Whenever you study for these, right? Whenever you study the antibiotics, there's depending on how prepared you are in this field or in this topic. I'm assuming a lot of you have never studied this before or just know like the basics. Try to find something unique about every class. Try to figure out how this class stands out from the other one, and that will help you kind of maintain, um, I guess, some sort of order in your head. But at the same time, try to link it to other classes that you already studied. So, like with glycopeptides, like I told you, remember that it's acting on the same I guess, not organelle, but on the same molecular structure as the beta lactams. So that's what unites these two families, but at the same time, it acts on it in a different way, in a different fashion. All right. All right, the next class, vast class of antibiotics that we will look at are the antibiotics that inhibit the protein synthesis. So the translation. Um, so whenever you hear protein translation, whenever you hear translation protein synthesis in bacteria, you must remember that bacteria synthesizes proteins a little bit differently than how we do it. And that is, again, the foundation for selective toxicity of antibiotics, right? Because if we had the same mechanism, then these antibiotics would be just effective against us as they are against bacteria. So that would be bad. Remember that bacteria synthesize the proteins, translate the proteins using a different types of subunits in their ribosomes. They're using 30S and the 50S subunits. We don't have these subunits. And most of the action of these antibiotics will be on these subunits. So they act on the subunits, and one class of the protein synthesis inhibitors specifically arrests, stops elongation of the protein chain. So it prevents the elongation of the protein chain, and we will discuss separately how it happens. So protein synthesis is inhibited. So what kind of representatives, major families of antibiotics that are part of this category, of this group? So the first one we're going to look at is the aminoglycosides. Pretty sure this link doesn't work as well. I'll just explain it. So the aminoglycosides, they bind to 30S subunit of the ribosome. Okay, they bind to the 30S subunit of the ribosome, and as a result, actually, let me check. This one might be available. No, they were from, some of them were from different sources, and some of them could be active, but no, it's not. All right, so by attaching to the 30S subunit of the ribosome, aminoglycosides quite often just cause incorporation of the wrong amino acid in the sequence of the protein. Okay. Uh, in some cases, they actually cause the rest of protein synthesis uh, um, by binding to the 30S subunit of the ribosome. So the protein synthesis is stopped, and the whole protein that is made is becomes a so-called truncated protein, a shorter version of the protein than it should have been, right? Truncated. Those truncated proteins, because they're so much shorter than the original, the wild type, so to say, copy of that protein was supposed to be, those truncated proteins are incapable of performing their intended um, functions. So that protein is essentially lost for the cell, and the cell quite often uh, ends up dying without those proteins. Those proteins usually are performing some of those proteins will inevitably be performing some essential roles for the cell, and if they are truncated, if they're just, it's like think of it as I don't know, like a broken key, so to say, right? You had a long key with you know, uh, the pattern on it that should fit the lock, and then all of a sudden half of the key has broken off, right? So obviously you won't be able most of the time to open the door with such a key. So that's what happens with these truncated proteins. They are bactericidal, so they actually kill bacteria. And again, the reason they kill bacteria is because they create these proteins that are incapable of performing their function. And the cell, as a result, that cell lacks significantly, severely in a certain functionality as a result of that. And if that functionality, for example, has to do with energy harvesting by the cell, for the cell, obviously bacterial cell will not be able to survive when such proteins are made. So aminoglycosides are bactericidal. They can be toxic sometimes to people. They may cause kidney damage if too much has been administered or if the person is sensitive. It is both active against gram negatives and gram positives, so it's a wide spectrum drugs, so they're effective that way. Uh, the reason why they're effective against both gram negatives and gram positives, you also don't just blindly memorize it. Make sure you understand why is that the case. This is the case because these two types of cells, gram negatives and gram positives, their protein synthesis mechanism is exactly the same. There's nothing different about it. Unlike your gram negatives and gram positive cells, having different structure of the cell wall and the cell membrane. Protein synthesis is exactly the same, so the aminoglycosides will be efficient the same way against both, equally efficient against both classes. Okay. Major representatives: neomycin, very commonly employed antibiotic for topical use. If I'm not mistaken, the neomycin is an antibiotic that they add to another one of my favorite quote unquote products. I don't remember if I mentioned this earlier to you or not. It's the cream that they sell. Um, Forgot what the name of the cream is, but whatever. You probably know what I'm talking about. The cream that is being applied to the skin. Whenever you get a cut or something like that, you apply it to your skin, and that cream actually has neomycin antibiotic. Again, very controversial product because you don't necessarily have to expose your wound to antibiotic right away. Unless you're immunocompromised, unless you have severe issues with immune system, and unless your cut that you obtained is like really bad, you don't need antibiotic on that wound. If it's a simple cut, you just have to kind of apply, uh, like stop the bleeding and then let the body heal the wound. There's absolutely no need to kill bacteria or force bacteria to mutate in response to these antibiotics. So people still use it, still being sold. People think it's very safe for them to use. I mean, in some cases, it's not. I mean, it's not going to hurt you in a way, but right? But at the same time, like I told you with antimicrobial soap, you are on a large scheme of things, you're promoting resistance in bacteria. That cream ends up in the soil. That cream ends up in the runaway water or runoff waters, and so on and so forth. So mass usage of antibiotics in products like that for like casual use when you like cut yourself or whatever, it's not it's not a good idea. It's not a good idea. Gentamicin, another antibiotic. Gentamicin has limited value. It is good. It's still, I think, being employed against Pseudomonas species for people with cystic fibrosis. Very effective against those Pseudomonas species. And gentamicin is also um, very widely employed in research. You know how in recombinant DNA you were isolating and transforming the plasmid that was carrying ampicillin resistance on it, a blood gene, right? BLA. By the way, the BLA gene refers to beta lactamase gene, right? BLA stands for the beta lactamase, and that was the ampicillin resistance gene. So the same way as the people use ampicillin in research, they also use gentamicin. Very, very common drug in research. Gentamicin, ampicillin, um, forgot the third one, the biggest one, but we'll see it in the lecture slides today. As far as using it outside of cystic fibrosis patients, it's not very common antibiotic these days. It's not. There are better options. Tobramycin, also useful against Pseudomonas, also being used for cystic fibrosis. As far as memorizing these specific ones, don't need to. I'm not going to be asking you which of these three belongs to aminoglycosides. This is just to illustrate it. But do remember everything about the mechanism of action. Tetracyclines. So the tetracycline is also a different family of antibiotics, but they are still protein synthesis inhibitors. So that's one of those cases where multiple classes of antibiotics target the same mechanism, but they target it from a different angle, right? A different mechanism is employed. So in this case, tetracyclines is also a tetracyclines are broad spectrum antibiotics, again, for the same reason, because gram negatives and gram positives have no differences in the way they synthesize the proteins and the way they translate the proteins. It was actually extracted from the streptomyces that I told you before about. And the way these act, they block the attachment of the amino acid that is carrying the tRNA. The tRNA that carries the amino acid, they block the attachment of that charged, as they call it, tRNA to the 30S subunit of the ribosome. So the new amino acid just doesn't get incorporated into the growing peptide chain. So elongation doesn't happen. And if elongation doesn't happen, you also generate so-called truncated, too short of the proteins. The proteins that are too short to perform their proper function. Okay, so they are also bactericidal. As a result, the cell is lacking functional proteins. Quite often, those proteins are essential for the cell fitness, and as a result, the cell dies. So they are bactericidal. One, I guess, spectacular and not so much feature of tetracyclines that you might memorize them by is the fact that these antibiotics, tetracyclines, were still are being used a lot as animal feed additives. So for the farm animals, goats, whatever, chicken, cows, and so on and so forth, pigs, they use tetracyclines to feed them antibiotics. As you know, actually, one thing I want to take a note of that. I'm not going to be asking that on the test, but it's also one of those fun facts to remember. Whenever we talk about antibiotics resistance and how, we as people, as humanity, are selecting for antibiotics resistance, people usually talk about the hospitals and how we abused or use a lot of the antimicrobials in the hospitals. How people are using antimicrobials outside of hospitals, all these disinfectants and whatnot. But the truth is, the interesting fact is that About 75% of all antibiotics that we generate, that we produce industrially, about 75% of those antibiotics actually go towards animals and not to people. Only 30 and 25%, 25 to 30% of all antibiotics that we generate are being used by people. Everything else goes as animal feed additives. So the selection for the resistance happens there in the farms, in the soil, in the water as well on a large scale. Um, Obviously, this was severely abused in 20th century once people realized that you can significantly increase the yield, meat yield, all sorts of yields of your farm animals by using antibiotics, by keeping them alive, by keeping them healthy. And that obviously leads to uh, a significant reduction of price of meat, right? That is generated on farms like that. Without using antibiotics, that meat, that those animals, that production would be a lot more expensive. Would be a lot more expensive. Um, Speaking of the claims, when people say, "Well, let's just get rid of all the antibiotics," kind of to wrap up this topic a little bit, let's get rid of all the antibiotics that we give to animals and so on and so forth. Uh, it's a very brave and like emotional claim that quite often everybody uh, feels like it's a good claim to make. But at the same time, if you think about it, if you were to get rid of all the antibiotics um, administered to farm animals, think of the prices, right? How affordable that meat is going to be now for the general public. The price will obviously skyrocket. So there's always pros and cons. They do select for resistance, obviously, on large scale, but at the same time, it makes food a lot more accessible to majority of population. Some of the disadvantages of the tetracyclines, again, some of them maybe unique specific features. They're broad spectrum antibiotics, very efficient, bactericidal, like I said. They're pretty good, unfortunately, at suppressing gut microflora. And we talked about that before. I don't want to repeat myself. Very important for our immune system and in general for our health and well-being. So tetracyclines are not the best antibiotics we have. They're not the most commonly employed antibiotics we have in hospitals for people. And that is why, one of the main reasons why is because they are very good, pretty good at suppressing gut microflora. Another thing is, another major problem with tetracyclines, why they were phasing them out over the past decades, is the fact that they may cause severe adverse effects in bones, especially when these antibiotics are being administered to kids when their skeleton is just being formed. So they basically deposit themselves in bones, these antibiotics, and they compromise the structural integrity and strength of the bones. So that can be actually very detrimental. They can also deposit themselves in teeth, especially like I said, when it's being given to kids. And sometimes you can like see people or used to see, I guess, more in the past, people with like yellow teeth, like severely yellow teeth. That means most likely that they were administered tetracyclines in their early childhood. So they're being phased out. There are better antibiotics out there. Depends on the region of the world, obviously. There's a lot of reports of really bad antibiotics being used in developing countries, the ones that are not used anymore in Canada and States. But usually those ones are just cheaper. They can also cause liver damage in pregnant women. So pregnant women always have higher sensitivity to all these xenobiotics, all these drugs, and specifically liver damage from tetracyclines is known for pregnant women. So again, not the best antibiotics, not the worst, not the best. People try to avoid them using them these days. The next category, I guess, the macrolides. Still, again, pay attention, right? How it's still the drugs that are inhibiting the protein synthesis, just on a little bit different, um, from a different angle. These are some of the representatives. This is the formula of these macrolides, antibiotics. You don't have to memorize the formula. The reason I put it up there is to show you, to draw your attention to one property of these antibiotics. These antibiotics are very bulky in terms of their 3D shape. They're very big molecules. Okay, and this will have one interesting consequence to their mode of action. So, how do they work? How do they actually exert their effect? They prevent the elongation of the peptide chain, but not the same way the tetracyclines were doing it. So, they actually block the activity of peptidyltransferase activity. It's the enzyme that, as you should remember, the enzyme that is responsible for linking a new incoming amino acid to a growing amino acid chain, right? So the enzyme that is responsible for putting the amino acids together and forming that growing chain of polypeptides. So if the new amino acid is not being attached to the existing chain of amino acids, obviously your peptide ends up being also truncated. It ends up being too short, right? So peptidyltransferase activity is blocked, and as a result, the translocation of the ribosome is also blocked. So the ribosome doesn't even sometimes move into the exit position. So the peptide chain may not even be expelled from the ribosome. So all in all, you're getting severely compromised peptides in terms of their structure. They are unable to perform its proper function, so they are bactericidal as well. One thing about these ones, the unique feature, the feature by which you should remember these antibiotics. So you would totally expect, based on what I explained already, that since these antibiotics are inhibiting the protein synthesis, and the protein synthesis being the same in gram positives and gram negatives, you would expect these antibiotics to be equally effective against gram positives and gram negatives, just like tetracyclines were, just like aminoglycosides were. But with macrolides, it's a unique story. They are mostly effective against only gram positives. Very selected few gram negatives are susceptible to macrolides. So the question is, what's going on? Why is this the case all of a sudden? It's not like peptidyltransferase activity and translocation of the ribosome is any different between gram positives and gram negatives. It's not. It's the same. So why is this the case? This has to do with what I already mentioned to you earlier, the size of the macrolides. They are so big in their size and their 3D structure that they just are unable to go through the double layered cell membrane of the gram negatives. As simple as that. Gram negatives just do not allow these antibiotics inside of the cell because of the double layered cell membrane. Two layers of cell membrane is a very efficient tool at keeping chemicals away from inside of the cell, especially when that chemical is such a huge molecule as macrolide. And that's why only gram positives, or mostly gram positives, are susceptible to macrolides. If somehow macrolide manages to get inside of the gram negative cell, it will be just as efficient as it is for the gram positive cell. It's unable to penetrate the two layers of the cell membrane of the gram negatives. Okay. So whenever they tried, as a result of that, you would imagine the semisynthetic modifications of macrolides, they try to give them better penetration towards gram negatives, as well as better tissue penetration. Because of their such a huge bulky structure, they're also very bad at penetrating tissues. And that is obviously bad for drug delivery. the drug doesn't end up where it's supposed to be. So semisynthesis tries to make these drugs more um, effective in terms of penetrating tissues as well as gram-negative cells. Okay. The next class, major family that will now have a different mode of action. Now we're finishing with DNA and RNA synthesis. Sorry, with the protein synthesis, we are now looking at the antibiotics that uh, affect the DNA and RNA synthesis. And we only have like a couple of slides remaining, so let's make a quick break, like 5 minutes break, and come back at 8:42 and finish this lecture. So I'll see you in 5 minutes. All right, we're back. Let's wrap it up. So this family inhibits the DNA and RNA synthesis, um, and one of the I guess yeah, one of the groups of the antibiotics we'll look at is the rifamycin family. So rifamycin family inhibits specifically mRNA synthesis. So it inhibits transcription. Make sure you know that. I might be using, I might be formulating the question such a way where you have to know what, have to rather remember what transcription and translation is. You've studied this before. Rifampin is, I think, the only representative we will look at. Or actually, yeah, I think it's the only one. So it's a very good family of antibiotics. It's very effective. Uh, Acts against a lot of problematic species, just like mycobacteria, tuberculosis, in the treatment of tuberculosis and leprosy. But there's one major problem with this particular family, as well as with the other family that inhibits the DNA synthesis. These antibiotics are very easy to develop the resistance against. And this resistance mechanism develops through a mutation in RNA polymerase. RNA polymerase mutates. It's a very simple, very commonly bacteria. Let's put it this way, without going too much into the details. Bacteria is very um, what's the right word? I guess they're very good. They're very quick at mutating their RNA polymerase. And when they do so in large quantities, sooner rather than later, you will find the mutants that will be unable, that the rifampin will be unable to bind to. And as that happens, obviously that mutant becomes resistant to the antibiotic, and um, it cannot exert its effect. Therefore, bacteria is able to survive. So that's probably the only major problem with this class of antibiotics: quick development of resistance. A lot of valuable characteristics, able to penetrate tissues very well, reaches high therapeutic levels in cerebrospinal fluid. So all of these categories are good. All of these properties are good, but unfortunately, resistance can develop very quickly. It still is being used. It's just another class of antibiotics, quinolones and fluoroquinolones. These ones inhibit the DNA synthesis, not RNA synthesis, but DNA synthesis. Quinolones are the um, base molecules, so to say. Fluoroquinolones are the semisynthetic analogs of the quinolones. So they take quinolones and semisynthetically modify them to make them fluoroquinolones. So what are those? What are those antibiotics? You actually encounter them quite often. It's a very common antibiotic prescribed by doctors. Ciprofloxacin belongs to fluoroquinolones. Norfloxacin, nalidixic acid, especially nalidixic acid, I guess, is mostly employed for research purposes, research applications. It's not as good for us. But norfloxacin, ciprofloxacin are pretty commonly, especially Cipro is very commonly prescribed to people. It is one big benefit of these drugs. Uh, they're very good at treating UTI, urinary tract infections. So they're being employed a lot for that. So the way they act, they inhibit the DNA gyrase, um, specifically bacterial variants of these enzymes, topoisomerases 2 and 4, the ones that are not being employed by people, by humans. The DNA gyrase is the enzyme, as you should remember, the one that unwinds the DNA, and that is the process that is essential to happen before the synthesis can occur, right? So they inhibit that, and as a result, the DNA synthesis cannot occur. So these ones are bacteriostatic. So yeah, as it says here, often on antibiotics of choice against UTI, urinary tract infections, and pneumonias. The fluoroquinolones, the semisynthetics, are just having a wider spectrum of activity than traditional quinolones, and they're less toxic. Nalidixic acid is fairly toxic to people. Not the most toxic antibiotic out there, but fluoroquinolones are better. Same disadvantage as with um, the rifamycin family. The resistance develops very quickly because, again, the DNA gyrase is very easy, fairly easy for the bacteria to mutate. Once it mutates, and uh, there's a chance that that mutant will be unable to attach to, or rather, the antibiotic will be unable to attach to that mutant and therefore will not be able to inhibit the gyrase, and therefore bacteria will be able to replicate as it should. Okay. The last class of antibiotics that we'll look at, the sulfonamides, and these are actually those sulfa drugs that I told you about in the first lecture back in the day, and talked about a little bit today. So these are the drugs that were the first antibiotics that were mass produced. They were the first ones that were discovered. Those are the sulfa drugs, the ones that were discovered thanks to textile industry in Germany. They were using some of these derivatives of the sulfa drugs as uh, red dyes to the fabrics. They were dyeing the fabrics with these dyes, and then eventually they found out that they have antimicrobial activity. But if you remember, the problem with the sulfonamides was that people started dying, patients started dying in response to sulfonamides, and for some time they thought it was because of the toxicity of antibiotics themselves. But it wasn't actually the antibiotic, it was actually the excipient or the solvent that the antibiotic was being dissolved in. And they were using ethers, toxic to people, ethers, to dissolve these antibiotics. So, and in response to that tragedy, FDA in states was formed. We talked about it earlier. So the sulfonamides themselves, the sulfonamides themselves are, if properly used, if proper solvent is being used, are very good antibiotics. They are still being employed, and they're a little bit unique in compared to the other classes that we looked at today, in a way that they don't act on any molecular processes or molecular organelles inside of the cell. They don't affect any enzymes. I mean, molecular enzymes. They actually affect uh, metabolic pathways. They affect chemical reactions. Chemical reactions. So the metabolic pathways are a set of reactions that lead to the production of a certain important biomolecule. In this case, sulfonamides are acting on the pathways, metabolic pathways that are generating DNA and RNA nucleotides for bacterial cells. Okay, so if you look at this diagram here, you have sulfonamides acting on these enzymes. I will talk about it a little bit more in a second. You also see this guy here, trimethoprim. Trimethoprim, you need this. You need to know this for the test. This drug is always being looked at together with sulfonamides because it acts on very similar enzymes that sulfonamides do. Just a bunch of enzymes, as you can see, a little bit down the chain. But what you must remember, trimethoprim is not a sulfonamide. And here's the structure. This is the sulfonamide, and this is trimethoprim. Trimethoprim lacks the sulfur group that you have here. So structurally, it is not a sulfonamide, and it's not classified as sulfonamide. But it's always being looked at together with sulfonamides because of the similar activity towards similar enzymes. Okay, make sure you remember that. As far as their activity, what kind of enzymes they affect? They affect the enzymes that are responsible for the generation of dihydrofolic acid and tetrahydrofolic acid. These are the precursors for the DNA and RNA nucleotides. So as you can imagine, a very important pathways, a very important anabolic pathway for the cell. Anabolic meaning synthetic pathway, the pathway that leads to the production of the chemicals. Um, so how do they act? They inhibit the enzymes. They inhibit the enzymes that convert PABA to dihydrofolic acid, and then eventually dihydrofolic acid into tetrahydrofolic acid. So if the enzymes are inhibited, the concentration of these precursors, DHF and THF, is low, and therefore the production of DNA and RNA is severely inhibited. It is almost never completely shut down, the production of these precursors, but it can be severely inhibited. We'll talk about why it is almost never completely shut down when we talk about the resistance. But for now, just remember that. So it is significantly slowing down the production of DNA and RNA nucleotides. Now, the question to you, so as far as bacteriostatic versus bactericidal activity, if you are asked that question in the test, one way is just to blindly memorize what these antibiotics are. The other way is just to decipher this logically. If you inhibit the production of DNA precursors, DNA nucleotides, which, I guess, which process of the cell will this impact the most? The replication, right? When does the DNA replicate? When does the cell rather require new DNA nucleotides? When the cell is replicating. So by significantly cutting down on the production on those nucleotides, you are inhibiting the DNA replication, therefore the cell replication. Therefore, these antibiotics should be bacteriostatic, and they are. They are bacteriostatic. Very good antibiotics, actually. Very good at treating some severe UTI infections. These infections are on the rise in the past decades. We'll discuss this next week. So that is a good property to these antibiotics, that they are treating those infections. They're broad spectrum. Both gram positives and gram negatives have the same um, metabolic pathway at their expense to produce the precursors. Resistance is fairly common nowadays. I mean, it's fairly common for a lot of different drugs, but here you have multiple mechanisms of resistance that we will discuss next week, and that's why bacteria is fairly, I guess, masterful at being resistant, developing resistance to these antibiotics. We'll discuss how next week. Lastly, for these, something to think about, selective toxicity, right? My question to you, I'll answer it next week. You can think about it at home. You can look it up, but think about it first. So as far as the selective toxicity is concerned, how do these antibiotics, considering what kind of enzymes they inhibit, how do they not impact people? How are they selectively toxic only against bacteria? So what is it about us, about people, that makes these antibiotics not dangerous to us? So think about it. If you don't have the answer, you can always look it up. We'll talk about it next week.